So with the start of it, I want to give you a few reminders here. Okay. Last time, first, we spoke about this. Here, filtration, reabsorption, and secretion. And for now, we're going to focus on filtration. Remember when we talked about nephron? We mentioned that it consists of the glomerulus, which is essentially a bunch of capillaries surrounded by a capsule, and nephron tubules, proximal convoluted, distal convoluted, the nephron loop or loop of Henle. We started to look at the processes that happen during filtration, and essentially the name of the process reflects what's going on there. Do you have filters at home? Did you ever use filter? Those of you who don't have filters at home. So it's basically a really tiny sieve, right? So same situation is observed in the glomerular capillaries. First of all, we have three types of capillaries, right? We have continuous capillaries, we have fenestrated, and we have sinusoid. So these are fenestrated, which puts them in the middle position in terms of the permeability. Are we clear? And remember, now think about this. If you use the filter, do you have something like Brita filters? You know, when you pour water initially, and there's a lot of water on top of the filter, water drips fast. And if you put just a little bit of water, it drips slow, because the pressure of the water column on top of the filter determines how fast water flows through the filter. Does that make sense? So basically what I'm trying to say is that the main factor that normally determines the rate of filtration in kidneys is the blood pressure, which determines this parameter, hydrostatic pressure inside the glomerular capillaries, which is, as you remember, it's high. What is the reason for it to be high? Something about the size of arterioles. Well, it's a large afferent and small efferent. Does that make sense? Sorry. We're clear? Good. So basically it means that there's a lot of blood that flows into those capillaries, and much less blood, well, it's harder for it to leave, so pressure in the capillaries is high. Okay. Normally, well, this parameter, hydrostatic pressure in the Bowman's capsule, is fairly standard, so we're not going to disregard it, don't get me wrong. We're just not going to look at it as the factor that can influence the filtration, unless you tie somebody's ureters completely and lead to the accumulation of urine in the kidney. Does that make sense? I mean, you're not going to do it, but hypothetically, the blockage of complete obstruction of ureters may lead to accumulation of filtrate in the kidneys. Okay, but that's kind of an extreme situation. So this factor, osmotic pressure, capillary osmotic pressure, that actually draws fluid back from the capsule into the blood, basically depends entirely on the concentration of plasma proteins. Does that make sense? Now, if you would look at these three pressures, you will see that one pressure that one could say, is possible, isn't there. osmotic pressure that drives fluid into the capsule is not there, just non-existent normally in the normal kidneys. Why? What is normally absent in the filtrate? Which molecules that sort of produce osmotic pressure and are present in plasma are not normally present in the filtrate? Proteins. Does that make sense? So proteins do not normally present in the filtrate. How does that happen? For this we need to figure out the structural elements of the glomerulus that are responsible for actual process of filtration. This picture in the right side of the slide, you can see a capillary, the red structure with the multiple fenestrations, surrounded by podocytes. Can you see them, podocytes? So each cell, I kind of loosely bounded podocyte. So podo means foot, okay, and these processes here, these processes are called foot processes, the podocytes. Imagine them being somewhat like amoeba cells, spreading the foot processes all over the capillary wall, surrounding the capillary from all sides. Does that make sense? Do you see that here? These foot processes, okay, that surround the capillary. Now, between these foot processes are so-called filtration slits. You can see the little slits, and those slits, in fact, determine the process of filtration. Am I making sense here? So think about this foot process as making a sieve. The size of the filtration slit is smaller than the size of the proteins. Essentially, filtration slits allow the passage of only rather small organic molecules, such as urea, glucose, and amino acids and drugs and whatnot, but prevent proteins and, of course, cells that can be found in the blood to cross the filtration membrane. Does that make sense? So essentially, things that can leave the glomerulus, and enter the capsule will be water, ions, and small organics. We're clear about this. Now, let's go back to the filter. So if you have a filter, can you break it? Can you rupture a filter? Yeah, how? How can you rupture the filter? Too much pressure, right? So it turns out high blood pressure can damage the filtration membrane here, can damage the podocytes, can damage the filtration slits, leading to the appearance of protein in the urine. It's basically first sign, the presence of protein in the urine is the first sign that something is wrong with glomerulus. It can be a blood pressure induced damage, or it can be the result of infection, but it means that something is wrong there. We're clear? Now, before we go on and chat about the kidney function and glomerular filtration, right? Well, before we talk about kidney function and all the regulatory features of the kidney, we need to define what is the glomerular filtration rate is. GFR is the volume of filtrate that is formed in the kidneys in 1 minute. So every minute of your life, your kidneys form from 120 to 125 milliliters of filtrate. Does that make sense? So I'm trying to calculate. Yeah, it comes up to about 180 liters of filtrate a day. Now, this should give you a clear understanding of the difference between the filtrate and the urine. How much urine regular person produces a day? From 1 to 1.5 liter, approximately. In the same period of time, an average person produces 180 liters of filtrate. So one of the kidney functions, let's write it down. Urine, you don't need to memorize the number, 1.5 liters. Filtrate, 180 liters. What I'm trying to tell you is that your kidneys not only produce filtrate, they also concentrate it incredibly well, because the majority of this 180 liters is water. It would be not really feasible to drink a comparable amount of water a day to not die from dehydration. 180 liters is a lot. Okay? That makes sense. Now, what determines glomerular filtration rate? 3 main things. First, net filtration pressure. Second, permeability of the membrane. And third, surface area. So let's start to figure out each of these factors one by one. Membrane permeability, which means the ability of these podocytes and capillary endothelium to let things through. Okay? This normally membrane permeability is really high. Just it's so high that we don't take it into account. It's not really a variable here. It works perfect. Okay? So we knocked it out. Now, surface area. If you would look at this image, you will see that glomerular capillaries are really constantly bending, you know. So, surface area for the volume of the glomerulus is huge. Are we clear? However, it can be regulated. Okay? There are so-called mesangial cells that can basically tighten up capillaries and reduce the surface area. Are we clear? And finally, net filtration pressure. Now, let's take a look at the components of net filtration pressure. Right here. Let's knock them one by one. Do we really dramatically change the concentration of albumin in the blood, or do we not? Normally, in homeostatic conditions, amount of protein in the blood is what? Changing or not changing? Not really changing. Does that make sense? Which means this parameter, the osmotic pressure, is not going to be regulated. We're clear. Unless you put some clips on the ureters, unless you completely block the urine outflow, hydrostatic pressure in the Bowman's capsule is not going to change. We're clear? Essentially, the main regulator will be net filtration pressure, will be the hydrostatic pressure in the capillaries, which reflects which parameter in the cardiovascular system, huh? Blood pressure. Now, if you increase pressure on top of your filter, how does it change that? How does it change the filtration rate? Increases. That makes sense? Are we clear about this? Now, kidneys are somewhat unique because they participate in two really important homeostatic processes, homeostatic regulatory processes. One is to regulate their own function. And a kidney function is basically determined by GFR. If you ever encountered patients with chronic kidney disease, what is the parameter that is measured in those patients? GFR, right? It's glomerular filtration rate. And in kidney disease, it is decreased. Basically, drawing the analogy with, say, industrial or household filter, it means that filter is clogged. Does that make sense? Somehow it is not really getting the job done. Does that make sense? We good? So kidneys by themselves try to maintain normal GFR. Any change in the glomerular filtration rate in the kidney will be met by homeostatic changes in the kidney to bring it back to normal. So if it goes from 120 to 140, there will be a response to bring it back to 120. If it goes from 120 to 100, there will be a response to bring it back to 120. Does that make sense? So why normal GFR is critically important. Normal GFR, normal glomerular filtration rate, ensures that the filtration process is also normal, which means the concentration of ions in extracellular fluid is normal. Is that clear? There's no acidosis, there's no hypokalemia, I don't know, hypercalcemia, whatever. Pick your own favorite term. We good? Now, the problem is that the whole body has another parameter that it has to regulate by kidneys, the blood pressure. Does that make sense? So if blood pressure decreases, then the immediate response is to so filtration through the kidneys decreases. When filtration through the kidneys decreases, how does it affect GFR? It also goes down. Okay? Now, technically you can say, okay, but kidney response will be to increase GFR, to increase the filtration rate, which will negatively affect blood pressure. So it kind of comes to the confusing situation. What is more important? Constant GFR or normal blood pressure? It turns out that in the range which is outside of more or less normal blood pressure, extrinsic controls that regulate blood pressure will dominate over intrinsic. Does that make sense? Do you understand what I'm trying to say? Think about it this way. You have a corporation, okay? You have a corporation, and this corporation consists of many branches. And if everything is good, everything is running well, then, I don't know, every branch has a certain degree of freedom in what they do. They may run some customer promotion event, you know, they may spend some more money on this, some more money on that. But if the market is really tight, okay, or the finances aren't looking really good, then everybody's going to be tightened up. You know, we have to fire like 50% of your employees. Now we don't really care if you like them. We just need to make those cuts, okay? Is it going to hurt the business? Yes, it's going to hurt the business, but we can't afford. So this tight market is the situation when blood pressure is either extremely high or extremely low. Does that make sense? So these intrinsic controls, as I mentioned, they maintain glomerular filtration rate. Extrinsic controls maintain blood pressure. And it's a constant teeter-totter between those two. Okay, and we're going to talk about how they work. So first of all, general idea. How does intrinsic control mechanism work in general? So imagine there is a slight increase in blood pressure. Say you drank a gallon of water. Increased blood volume, increased blood pressure. How does it affect GFR? It immediately increases, right? Which immediately leads to the response from the kidney to reduce the blood flow through the glomerulus. When blood flow through the glomerulus decreases, GFR decreases back to normal. That makes sense. Now, extrinsic controls will regulate blood pressure in the way, so this is the direct regulation. Blood pressure goes up, blood flow through the kidneys increases, rate filtrate production goes up, amount of urine that is produced goes up, volume of blood goes down, blood pressure goes down. Does that make sense? So intrinsic controls will participate in minute-to-minute basically regulation, like small adjustments to varying blood pressure. Extrinsic controls will be responsible for more robust, more systemic changes. Now, if you think about this, even relatively small changes in the blood pressure may dramatically affect GFR. Okay, one of my favorite examples actually is this. So let's say systemic blood pressure is, let's make it 100. Okay. Now, if your systemic, I'm talking about mean arterial blood pressure. You decrease it by, forget about 80, 20%, from 100 to 80, which is not horribly low. Okay? It's going to be, say, even not 90/60, it's probably like a little bit higher than that. Okay? We decrease it like that, 90/70, for instance. Decrease it like that, the glomerular filtration rate will go down, hydrostatic pressure in the capillaries will go down, and it needs to go down by about 20%, like to 44, to stop any filtration through the kidneys. Does that make sense? So this fairly moderate decrease in blood pressure will lead to the zero filtration. Why zero filtration is bad? Why it's so bad to have GFR at 0? You're not getting rid of anything, If you experience, I don't know, hypercalcemia, or worse, hyperkalemia. Okay, you cannot get rid of potassium because it doesn't even make its way into the tubular system of a nephron. There's no way to regulate it. Does that make sense? So you can see that, it's kind of double-edged sword, and in case, there is need to regulate blood pressure, we will have to sacrifice kidney function. Now, how can... Yes?
So, for the intrinsic control, the glomerular blood flow goes down. Is that to keep that filtration normal?
Yes. So, basically, now think about this. Like, you start to, run. Your blood pressure goes up. It's normal. Which technically should increase GFR, and it will. An immediate response from the kidneys will be to lower it, to prevent any possible damage to the podocytes, to basically bring back the homeostasis, because you are in the normal blood pressure range. Does that make sense? If you're out of that normal, quote-unquote, blood pressure range, then nobody cares about kidneys anymore. We just have to do something about blood pressure itself, because if it's too high, it's a brain damage. If it's too low, it's a shock. Okay? We're clear. I'm going to give you an idea about the mechanism. It's super elegant, really. So myogenic mechanism here. Myogenic. Controlled by muscles, right? When blood pressure goes up, how does it change? How does it influence the stretch of the blood vessels? Higher blood pressure leads to... Speak up, huh? Stretch of the blood vessels, okay? Now, this stretch is sensed by granular or juxtaglomerular cells here in the walls of the afferent and efferent arterioles. And in response to that higher blood pressure and higher blood flow, afferent arterioles will constrict, efferent will dilate, essentially bringing less blood to the glomerulus. Okay? Bringing less blood to the glomerulus and making the efflux of blood from glomerulus easier. Does that make sense? Essentially, if you imagine the glomerulus as a tub, like a bathtub, if the pressure is too high, you turn down the faucet and you open up the drain. Does that make sense? Now, if pressure is too low, stretch is low, you dilate afferent arterioles. You dilate afferent arterioles. And you further constrict efferent arterioles. So essentially, you're turning up the faucet and you keep sticking something into the drain. Does that make sense? That's, in my, it's super elegant. Now, you can imagine that this responds to small variations in the blood pressure. Does that make sense? Now, so-called tubular glomerular feedback mechanism is also super elegant, but there are more steps here. So there are three main types of cells that mediate it. Macula-densa cells in the walls of the nephron loop sense the concentration of sodium chloride. Granular cell, as I mentioned, they constrict and dilate afferent and efferent arterioles. Does that make sense? Do you see what I'm saying? And then mesangial cells here, I kind of colored the mesangial cells. They provide a communication between macula-densa cells and granular cells. Okay, so macula-densa cells, they sense sodium chloride. Granular cells regulate the diameter of arterioles. Now, look, this is super cool. Let's talk about high blood pressure. Blood pressure goes up. How does it change blood flow through the glomerulus? Increases. I'm going to ask really simple questions, believe me. So blood flow through the glomerulus goes up. How does it change the amount of filtrate that is formed? Goes up. Yep, it goes up. If more filtrate is formed here in the glomerular capsule, how does it influence the rate of the filtrate flow? It increases, right? Because more filtrate is formed. Now remember I told you about reabsorption. When ions are basically sucked back into the blood, if the filtrate runs faster, then reabsorption time will do what? Huh? Think about this. You are on the train. You have a platform. You have to jump back on the platform. If the train goes really slow, do you have a lot of time to jump on the platform? Is it easy? Now imagine train is speeding. I'm not even talking about bodily injury. You have much less time to jump on the platform. Does that make sense? So when flow rate is high, reabsorption time is low because those ions have very, very short period of time to jump back in the blood. Does that make sense? More ions stay in the filtrate, which means the amount of sodium chloride in the filtrate goes up. Does that make sense? This is sensed by macular-dense cells. Macular-dense cells signal to granular cells to constrict the afferent arteriole. When afferent arteriole, this one, constricts, what happens to the blood flow through the glomerulus? It decreases. The amount of filtrate goes down. The flow rate of filtrate goes down. Train runs slow, and those ions have more time to jump back into the blood. So reabsorption time increases, reabsorption increases, and less sodium chloride is now in the filtrate, which I think is pretty awesome. Now, before I let you go, a few things. In this chapter, there's going to be a lot of multi-step negative feedback regulations. So you need to know myogenic mechanism. Basically, step by step. Like I tell you, if blood pressure goes up, what happens? You tell me, stretch increases, GFR increases, afferent arterioles constrict, efferent dilate. That makes sense. And same goes for tubular glomerular feedback mechanism. Okay, so basically my suggestion, draw it as the flow chart. I'll see you on Wednesday.